molecule, and this molecule is a division inhibitors. It did not allow DRP1 to act like a scissor to cut mitochondria. So then this MDB1 was introduced before the administration of NSAID to the rat. So then after exposing rat to NSAID, then we expose the stomach and analyze the biochemistry of mitochondria to follow it. Interestingly, blocking and aberrating nutrition largely prevents NSAID-induced mitochondrial metabolic failure. So if we block these abnormal mitochondrial fusions by using division inhibitors of mitochondrial fusion, can restore NSAID-induced abnormalities in gastric mucosa. Now the question is that, again, we tested macromolecular oxidations, cardiolipin content, caspase 9 activity, caspase 3 activity in presence and absence of these division inhibitors. Highly interesting that this is prevented. Now the question, MDB1 prevents NSAID-induced DRP activation, ubiquitinations, and pro-inflammatory gene expression also. That there is not allowing mitochondria to go for signaling pro-inflammatory pathways to damage gastric mucosa. Now the question is that, then again, we looked at by doing SORET spectroscopy that it certainly prevents gastric ulcer or not. That is the ultimate goal. So that MDB1, even in presence of NSAID, can effectively block gastric mucosal ulcers, as evident from the SORET spectroscopy and as well as the histological analysis of the gastric mucosa. And the middle panel is NSAID induced. In presence of division inhibitors, you will find no black spot in gastric mucosa. So this is a really very good tool by which we can bypass proton pump inhibitors or histamine H receptor blocker. But this is a model molecule. This is not a drug. So now this type of molecule can be utilized if we sensitize the pharmacologist or organic chemist to synthesize this kind of molecule. Now the second task that I don't like to allow the stomach to be damaged, and I'd like to take, I'm interested to absorb or continue with the NSAID. So then the next target, the level 2, that's the mitotempo. It's a beautifully designed molecule, and this is a nitroso compound. And this compound, what happens, it can only go inside mitochondria, and thereby going inside mitochondria, it can trap superoxide. Because my proposal was that blocking of complex 1 allows electron leakage from NADH not to go to meet molecular oxygen to the water. And then what happens if I use mitotempo, mitochondrially targeted nitroso compounds having the capacity to scavenge superoxide specifically. So this will really give some information that it will be effective against NSAID. So this was exposed just like MDB1 before oral administration of non-steroidal anti-inflammatory drugs. Then I opened the stomach and then did the experiment. Interestingly, mitotempo concentration dependently blocked the injury index as produced by gastric mucosa by indomethacin. And you look again the story of it, and you know the SORET spectroscopy, the blue one is the bleeding. In presence of mitotempo, the red and green one is reduced. There is no bleeding at all. So by only observing, because microscopic histological experiments may guide you something wrong information. So better to use the number of heme proteins coming out the mucosa by SORET spectroscopy that will guide you the exact quantification of the leaking of hemoglobin. So by this way, you can judge. So in this way, then again we tested the classical ways. The mitotempo corrects NSAID-induced mitochondrial metabolic defects. What are those? The defects induced by mitochondria: macromolecular oxidation, glutathione estimation, cardiolipin, mitochondrial potential, fatty acid oxidation, dehydrogenase activity, electron transport chain activity, and mitochondrial respirations. All the parameters, the mitochondrial health index is being judged in presence of this molecule. What happens? Mitotempo corrects NSAID-induced mitochondrial bioenergetic crisis, inflammation, and apoptosis. What happens? Now the question you may argue that, of course, you are giving ornamentation to your work, but how can you exclude the possibility that because of blocking of acid secretion, that is the reason for your prevention of gastric ulcer? So then what will happen? This is a beautiful molecule because the whole world is thinking not to block acid secretion, but to prevent gastric mucosa. If you block acid secretion, so then the gastric microbiome will be very much effective and will invite a lot of neurodegeneration, cardiomyopathy, a lot of problems. So if you block by proton pump, so then there should not be any acid to be secreted. So protein will not be digested. It will be fermented, and there's an outgrowth of different gut microbiota. So by this way, we are looking a molecule which will prevent gastric mucosa but not blocking acid secretion. So this is the molecule, by doing all this measuring gastric pH and acidity, both MDV1 and mitotempo did not block. So the question is that the pathological signaling targeted or triggered by NSAID impinging into the mitochondria, the story is that NSAID triggers superoxide to accumulate within mitochondria, causing mitochondrial oxidative stress and biology crisis. Mitochondrial oxidative stress hyperactivates PKC zeta P38 GLP-1 axis, which in turn leads to the mitochondrial fragmentation. Fragmented mitochondria initiates gastric mucosal cell apoptosis, blocking mitochondrial hyperfusion, protects against NSAID-induced gastropathy. Selective scavenging of mitochondrial superoxide by mitotempo protects against NSAID-induced gastropathy without affecting acid secretion. So far, I can tell you this is the story. Now I'll go to the real work right now. So this is a fundamental work. We established that. I'm not happy. So the next I will tell, NSAID causes severe gastropathy by damaging gut mitochondria. Mitochondrial oxidative stress and fragmentation underlie gastropathy due to excess intake of NSAIDs or painkiller. But still, there's an open question. Why mitochondria get severely damaged? What happens to the mitochondrial defense arsenal? I am telling you some observational studies, no mechanistic detail. Very observational, shallow studies I am presenting. Still to you. But the question is that what it does going to the body. So that was my next journey. I think they told me I can continue so that I can tell the story very lucid way. So then the highest technology right now in my hand is next generation sequencing, NGS. So we provide the control and NSAID exposed damaged tissue to NIDNG. Then we did series of transcriptome studies. And this will give you unbiased thinking that is there any culprit which is being switched on or off, leading to mitochondrial abnormalities. With this extensive heat map, data analysis, disease function involved, NSAID treatments, half gene networking, and we identified very marvelous way that is SIRTUIN-3, a molecular switch that is being highly reduced when it's exposed to non-steroidal anti-inflammatory drugs. Because this SIRTUIN-3, after venturing in details the morphology and its character, it appears that it is called mitochondrial guardian. It protects mitochondria. So that's why NSAID, by any means, affecting this HER3, thereby what we are reporting, I am reporting right now, what the downstream effects, the consequence of that. So this transcriptome studies, I am talking from very much in silico studies. Nobody will believe. So it needs validation. So with this data, we started doing wet lab experiments. What happens that transcriptome sequencing in rat stomach identifies HER3 as a target of NSAID. And what happens, HER3, the member of SIRTUIN family, class histone deacetylase, located in mitochondria, deacetylates free mitochondrial proteins, during the function of electron transport chain. So this is a very fantastic thing. With the progress of mitochondrial aging, mitochondrial proteins are hyperacetylated. If it is hyperacetylated, it will not continue its functions as vigor it was earlier. So a protein should be there who will deacetylate to make again to the normal form. So this HER3 is playing that role, thereby not allowing the protein to be acetylated. The acetylation clearing protein, and that's why called mitochondrial guardian. So this protein is being inhibited. So HER3 gene expression is started, and then NSAID depletes HER3 expression in vivo to trigger mitopathology. So we have observed that in presence of NSAID, there is a hyperacetylation of mitochondrial protein. Mitochondrial clearance of ubiquitinations, HER3 protein expression, mitochondrial DNA oxidation, mitochondrial integrity, and copy number. So we see all these things as instructed by the transcriptome and map. We started validating one by one. This story I'm talking about for more than 8 years of my research, for only understanding to venture some Cox independent target, because other scientists will argue with me, because I am creating a trouble with all of them. Because they are all happy with the prostaglandin, we are introducing. No, prostaglandin certainly, but it is not the final part. So by this way, what we identified that then kinetics of HER3 expression and gastropathy in vivo in response of NSAID treatment. So we are following the kinetics of HER3 expression and following the injury and its correlations, and then its substrate. So then recently, after doing all these things, we published in British Journal of Pharmacology very recently, and it is highly convincing that HER3 regulation is very fundamental for all. So then the question is that if SART3 reduction is the cause of NSAID-induced mitochondrial pathology and gastric ulcer, can it not be possible to upregulate SART3 in presence of NSAID? Then you will not get it. So we got a molecule called honokiol, a polyphenol from magnolia plants. So this is very popular molecule, and this honokiol is activating SART3 in vivo. So that I used for understanding and divulging in detail of it. Interestingly, SART3 stimulation prevents NSAID-induced mitochondrial damage and gastropathy. Again, transcriptome studies, which is being down-regulated by NSAID, is upregulated by honokiol treatment, which is upregulated, that is down-regulated by honokiol treatment. By this upregulation, down-regulation dynamics, we validated that SART3 upregulation by honokiol is preventing gastric mucosal injury. Now, what happens? Mitochondrial fragmentation accounts to GI pathology, that mitochondrial is fragmented, and because of hyperacetylation of mitochondrial protein, and there is a bioenergy crisis. So the three kinds of molecule can be exploited for better therapeutic uses, either develop honokiol-like molecules or develop mitochondrially targeted antioxidants, or a molecule which will prevent DRP translocations and preventing mitochondrial hyperfusion very selectively to gastric mucosa in neutral non-pH. So what happens? NSAID also kills. Now the question is that I have identified very nice thing, the toxic effects of a molecule. Now, how I can add values to this? So I like to kill cancer cell because this molecule is killing mucosal cells. So then if I kill gastric mucosal cells by NSAID, that is reported. NSAID is very much popular for because it kills pain as well as by inducing cell proliferations. So now the question is that my critic is asking me, the NSAID-induced cancer cell death is also associated with reduction of SART3. Or if I inhibit SART3 in cancer cell, then we can develop a new kind of immune therapy for cancer. With this thing, I started again. So this is called human gastric adenocarcinoma cells, and we did detailed transcriptome studies. And by these transcriptome studies, again we came to know by heat map and disease functions and hub-gene network, in human gastric cancer cell, if we add indomethacins or diclofenac or aspirin or whatever, so again it is down-regulating SART3 and induces the loss of cell growth. So what happens? As I propose that, then we go for detailed mining of NSAID serially down-regulates gene controlling metabolism and bioenergetics. You know, in mitochondria, there are proteins, 13 proteins. I used to talk everywhere in my lecture, totally preserved by mother, because mitochondria can encode only 13 very important complex proteins from their own genome, not borrowing from nuclear genome. Rather, it is coming from nuclear genome. So that's why, you know, if my mother's mitochondria is not good. So because in all my cells, I am having the mitochondria donated by my mother, not by my father. So my mother is providing me the energy for doing work. Father cannot. Father can give you only the nucleic acid. So this 13 protein is been encoded by mitochondrial genome, and it has been categorized by transcriptome analysis that NSAID is affecting these critical 13 proteins, thereby dismantling mitochondrial electrotransmission. It's a fantastic thing that, you know, by this, you know, the NGS is really a gold mine right now. It's NGS data because you have the highest performance. You can misguide you. So by this way, again, we validated this thing, dose and time-dependent reduction of SART3 expression in cancer cell. Now the final question is, being a biochemist, if it inhibits SART3, so why you not cloning and overexpressing a pure protein to test whether it inhibits or not? So then we overexpressed SART3 gene and first did docking studies that whether NSAIDs are really hooking to SART3 or not. It's fantastically, it interacts with SART3, and this clone overexpressed protein is tremendously inhibited by indomethacin and other non-steroidal anti-inflammatory drugs. So NSAID not only prevent the transcription or synthesis of SART3, it also inactivates already formed SART3 so that it cannot perform deacetylation in mitochondria. So by this way, we proved it. And then SART3 silencing. So then if SART3 is silenced, then low dose of indomethacin will work because it is inhibiting. So the synergistic effects we observe that SART3 silencing aggravates the cytotoxic effects of NSAIDs. So what happens? HER3 silencing aggravates the cytotoxic change again. So all mitochondrial proteins, mitochondrial potential, apoptosis, and fatty acid oxidation measures. Now I would like to conclude that the painkiller, which is inducing toxic effects to produce gastric ulcer by downregulating HER3, can be exploited the same target by downregulating in cancer cell to kill the gastric cancer. So NSAID is inhibiting gastric cancer by downregulating HER3. NSAID is inducing gastric ulcer by downregulating HER3. So the balance can be decided that how long, when, in what context, and the contextualization of the pharmacological activities will be fundamental pathways. And few things I'd like to advocate that we have some avenues to develop gastroprotective molecule bypassing the blocking of gastric acid secretions and hyperacidity. So thanks for your patience hearing. I'm very thankful to all of my students, Jessica's fellowship, DST, and my all students. Dr. Jhumur Ghosh is an instrumental in doing transcriptome studies, NIEMG. I'm that much not strong in transcriptome, but so she helped me a lot. Thanks for your hearing. Will you allow to ask a question?
Yeah. Okay, so normally in memorial lectures this doesn't happen, but the speaker himself.
So abnormally you can ask me a question.
So therefore I think it's fine with his permission.
Yeah, please. I'll be happy. I'll be happy.
Please.
Apart from the cytotoxic pathway, I think as far as I remember, in the 1998, another pathway has been discovered as the inducible nitric oxide synthase pathway. That is, the nitric oxide has found to be one of the regulatory molecules for the pain and everything. And I think there are 3 scientists who got the Nobel Prize to find out.
Murat, Ignaro, and they have actually got Nobel Prize by discovering natriuretic factors, nitric oxide. Yeah, true.
So, is there any molecule so far in the pharmacy or so far in the clinical trial phase where this molecule, which scavenges the nitric oxide, can be utilized as a painkiller apart from the cytotoxic pathway?
I doubt till my knowledge goes, because it is very non-selective. So, because that may arrest cardiac arrest, because vasodilation is necessary for nitric oxide, but you have to have generation of nitric acid specifically to gastric mucosa. It's a big problem, but there are avenues. But what I told you, I was very much unbiased, because I added non-steroidal anti-inflammatory drugs or treated either communicated cells to NIPMG or the whole tissue. Asking them, let me know, is there any clue for that? I don't like to make myself biased about this, that, because this is essential right now because of NGS data. Then if I get it, then you validate it. And again, I can tell you, nowhere, do not believe anyone. If one is talking, this is the molecule only targeting this and doing this thing. There is no such thing, I can tell you. Nothing. What we are using for aspirin to thin blood? It's not inhibiting prostaglandin. We are using anti-COVID molecule right now. So we are using right now so many things I can tell you. Say it's not possible. So what you have observed, we are all blind people, frankly speaking, describing the structure of an elephant. That I'm talking elephant is like a pillar. Someone is talking elephant like a big dove. Someone is talking, no, it's one kind of boiled egg. So why we are picking up, we are telling, because we are all perfectly blind persons still now. Yeah. So that's why, till date, based on my data, I can tell you, this is my suggestion. This I can tell you. Otherwise, I cannot tell. Then I will be orthodox and biased. I'm not at all a scientist. Because I am challenging Robert Vanny. He got Nobel Prize, so doesn't mean that he's wrong. He did something. Because of something, I am here. I am making some mistakes. Because of my mistakes, you ask me questions. So make mistakes. I can tell you out of my context one words. Swami Vivekananda told that if you make mistakes and start the whole world, and the whole world can establish to find the truth, then you are the generator and founder of that truth. Because of your mistakes, truth came in the world. So the question is that do something, try something out of box. Not who has told something. This is no. There's no sacrosanct in science. I can tell you. So
there is no such molecule as of now. that can actually increase the also directly clinical.
Yeah, probably. Absolutely. I think if you ask me some other question, so that getting any question, I can start.
There's just one more, just for the sake of time.
Yeah.
And I think there's just one more question from our co-chair.
Yeah.
Dr. Ghosh.
He is taking pantrazole, and what will be his fate? I am long.
Very layman's question.
Yeah.
Question is, when you are in tremendous pain, like I have back pain, I take diclofenac. From listening to your lecture, it seems to me that I should not do it.
No, I'm not getting frankly speaking. What you asking?
No, the question I'm trying to say is that for the painkillers are very popular because pain is something which is very difficult to withstand.
Right.
From your work, what would be the safe dose then? That is
Look,
the question.
from this work, what is very philosophically and scientifically, I can tell you, take NSAID, but simultaneously take not to allow mitochondria to be damaged. So, co-administration, that's why people are designing a molecule called ascorbic acid derivatives of Ranjak or some painkillers. So, in NSAID ran ascorbate, NSAID tempo, so that NSAID will kill pain. And the toxic effect is being mitigated by the co-administered molecules, not to allow
No, but that is still in the laboratory research, right?
No. It's mitocan. It's in the clinical features right now. Mitocans. Right now.
No,
Suppose you know why you are using pan D, pantoprazole D.
No doctor gives it now. When they recommend a painkiller, they simultaneously won't say that also take this. So you have to educate the doctors then.
No, that's why I'm giving lecture. Doctors are there. What should I do? So that's why I'm giving it my job being a scientist to get the story.
All right, thank you.
Pandy continues, is that what you said?
So Pandy, what Pandy is a product. Pandy means pantoprazole plus domperidone. Pandy means suppose someone is having not hyperacidity, it is small acidity, but it is not because of lack of peristalsis of the intestinal movement, it's accumulated. So domperidone is doing slickering the intestine, so that acid will go to the down. So pantoprazole is not effective in the Pandy. Just like indomethacin ascorbic acid is antioxidant, and indomethacin is painkiller. So the marriage, just I would like to tell this finish it 1 minute, you know all the story. When I was giving a lecture at my molecular immunology for one director asked me, so then I think I can tell you, you make a molecule by mixing an antioxidant with your NSAID. It must be. Then I told it may be, but must be cannot be. Then started I. So then I told, once upon a time an Irish actress asked George Bernard's song. So if you marry me, so then my offspring will be of your intelligence or my beauty. So the later, actually, if it is reverse. So the question is that, maybe I cannot tell you, being a scientist,
That was a Russian band.
Okay, thank you.
Hold on.
Thank you, sir, for such a wonderful talk. We now request the chairs to kindly present Dr. Uddhav Bandyopadhyay with the 4th Professor A.K. Sharma Memorial Lecture Award. Thank you, Dr. Ghosh and Dr. Ray, for chairing the session and doing the honors. To carry on with the session, may I now call upon stage Professor Shampa Das, INSA Honorary Scientist, Department of Genetics, Calcutta University, and Professor Naid Ali, the J.C. Bose National Fellow, INSA Emeritus Scientist at the Infectious Diseases and Immunology Division of CSIR ISCB. Ma'am, would you please come up on the stage and take over the session? Please sit. Now, Professor Shwampa Das and Dr. Nani Dani will be felicitated on the behalf of 21st AICCT. Ma'am, would you please carry on with the session?
So, good afternoon. I have an announcement. In the program, you have seen that after Professor Arun Sharma Memorial Award Lecture, there had been Professor G.K. Manna Memorial Award Lecture. But I'm sorry to announce that due to some unavoidable circumstances, Professor G.K. Manna Memorial Award Lecture today has been dropped, and it is not possible to present this award lecture today. But the conference should go, huh? Because it has begun and all the participants are here, so the conference is going on in its own flow. First, I congratulate the organizer for organizing this symposium on a topic, environmental toxicogenomics, ecosystem health and sustainability, challenges and way forward. So if you read this topic step by step, then you feel the importance of this topic. This is very contemporary topic, and I first congratulate the organizer for choosing this topic. Because every day, we are polluting our environment in various ways, knowingly and unknowingly. And environment is taking the toll of this threat. We are suffering from various diseases. Just now, you have heard a very nice lecture from Professor Uday Bandyopadhyay about the painkiller. Painkiller is people are taking regularly for anti-inflammation of their body, but this has tremendous ill effect. You have already heard from him. So in various ways, our environment is regularly polluted, knowingly but and unknowingly. But we have to take care of that. On top of that, global warming is taking the toll of environmental pollution. So this is the time how to protect our environment, because on this sustainability of the environment, proper environment, sustainability of human health, not only human health, all life forms of Earth is their sustainability is depending on the good environment. So our duty to protect environment. India, for the last few years, India is tremendously progressing in science and technology. There are lots of examples. I can present only one or two glaring examples, like COVID-19 pandemic. You see how our Indian doctors and Indian scientists have compromised with the COVID-19 pandemic. Compared to other countries, we have managed the problem so efficiently. Our doctors and researchers in record time developed several vaccines. And most recent incident, the remarkable incident that Chandrayaan-3. You see how ISRO has progressed in this technology. This is not only important in respect of space research science, but also it has far-reaching objective for protecting environment. They are trying to find out the water resources and the non-conventional energy resources from moon. So these are this way they are taking advantages. It is definitely India is progressing much faster rate, but still there are scopes to do much more on this. And in this era of genomics, so the topic is toxicogenomics is very important in this era of genomics, proteomics, transcriptomics. People had to find out how the environment is polluted and how scientists can find out the solution for protecting our environment. So see, our scientists are doing very well in this area, and in next 2.5 days, we will listen from different participants, distinguished eminent scientists and students, how they are looking this problem and how they are finding the possible solution from that. So here, first I invite Dr. Pritha Bhattacharya, one very renowned young investigator who is tremendously active in this area of research. So Pritha Bhattacharya, not only she is very socially conscious lady, not only she is a remarkable scientist in this environmental science research, but also I proudly announce that she is secretary of Kolkata Nivedita Shakti, and women-run non-government organization, Kolkata Nivedita Shakti. They are continuously working for the empowerment of underprivileged women and girl children to inspire them for study, science, and modern technologies, research, etc. So she is the secretary of that organization. So I am proud to announce that Dr. Pritha Bhattacharya is a wonderful woman doing science in one hand and also running for women enterprises. So please, I invite my co-chair, Dr. Nahid Ali, to properly introduce Dr. Pritha Bhattacharya.
So, hello everybody. Yeah, I'm given this pleasant duty to introduce Pritha, Dr. Pritha Bhattacharya. She is currently Assistant Professor, Department of Environmental Science, University of Calcutta. And her research interests, as pointed out by my co-chair, is in largely in epigenetics in the area of arsenic fields, nutri-epigenetics, non-communicable diseases like obesity and PCOS, trans metal toxicity, behavior epigenetics, occupational health, etc. And through her research interests, in the last 10 years, she has more than 70 peer-reviewed publications and experience in teaching in environmental health. And today she will be talking on, the topic is given there, arsenic, threats and challenges. With this, I welcome Preeta to give us the talk.
It's my privilege today. I was not a listed speaker of today's session, and somehow, just like we have had earlier the opportunistic diseases, but today I am an opportunistic speaker. So we are working in the field of arsenic toxicity for almost last 20 years during the days of my PhD. I hope I'm audible. I hope I'm audible. Last 20 years, also we are working on this. Also, last 20 years, we are working in this field of arsenic toxicity, but very alas, that last month we went to Nadia, Mahishwatan, the area, and I found that the people still, they are having arsenic contaminated water. The two different facts between, if we think that's the two perspectives, this 2 year short, just 2002 and now 2024, these two scenario, these two cross point, if I think the situation, what's the situation? Long back in 2002, when I have started my journey as a PhD student under Dr. Giri and Dr. Kunal Ray in this field, then we have found that people were not aware. Only in certain villages, some people had some skin lesions. They have dendropigmentation, palmar-plantar keratosis, and they had a stigma. They had a stigma that they cannot go and meet other, they cannot shake their hands, they cannot marry their girls to other villages. They were worried that what they are having and what will be the fate. But interestingly, also in a village of 100 people, if it's only 15 to 20 people, they are having this kind of lesions, while rest 75 to 80 people, they do not have any such lesions. And they thought that we are very safe. So from that, we have started our journey, that why, despite of this environmental similarity, environmental exposure, if we talk a lot about this environmental exposure, if it is same, why these different consequences are happening. And during that time, or still it is that World Health Organization, it states that depending on the skin lesion phenotypic presentations, we categorize the people as arsenicosis or other. So then we have started thinking or digging into that why there is this difference. And we are looking into the genetic scenario. So there must be some genetic variations. Nowadays we all know that arsenic is coming from the groundwater and also in the food. Just I was telling that in the last month when we went to Nadia, the people are saying that we went for this usual our medical camp, taking the water sample, urine samples, and the people are telling that, ma'am, we are buying the water from the outer sources. They have to buy the water. They don't have that enough money. And they are saying that we know that water will come one day. We will have pipeline connections. And 2024, maybe in the mid or at the end, we will get safe water. So where we are, that actually gives me a lot of pain. That despite of lot of research, lot of medical drugs, lot of things that we say, the genetics, the epigenetics that we are working, that we are just digging that when we are having this arsenic contamination through this drinking water or the cultivation, which we don't have any option. We are bound to have that whatever the food we are getting. So even in any part of the India, we don't know that from where the food is coming. So we can have that kind of risk. And these markers can be identified by the urine samples. These are the skin lesions, the plenty of camps, and they are aware. Unfortunately, they were aware with the fact they don't have safe alternative, and where we are failing again and again. To make the long story short, we made one prospective study that if the people are given fresh water or the drinking water that having no arsenic in their drinking water, what happens now? They had a long past history. So what we found that yes, if they are having safe drinking water with respect to arsenic, we find that their skin lesions reduce, their urinary arsenic concentration, if we measure, it's no more because urine is the current exposure marker, but the diseases we talk about, whether it's neuropathy, whether the other diseases, arsenic is a system toxicant. And it's such a nuisance that it affects all the organs. It affects our lung, kidney, whatnot, that are no more curing. The entire thing lies in between this arsenic metabolism. In arsenic metabolism pathway, it changes from the inorganic arsenic to organic arsenic. And this organic arsenic, there is MMA and DMA, which are actually excreted out through urine. So usually, earlier we used to measure the total arsenic concentration in urine, but with the advent of knowledge and the technology, we understand that it needs to measure this organic component. And what is the proportion of this MMA and DMA, that basically determines that what is the load of arsenic toxicity within our body, because what is the metabolism is going on. That's the important thing. And here, there is a lot of difference between the individual. So individual variation in this arsenic metabolism actually causes the change in the ultimate fate. So it indicates that although we have a similar exposure, if we have a difference in this metabolism, we can end up with a different result. And here, there are so many important things that come into. Arsenic 3-methyl transferase is one of the important enzyme which takes part in this entire metabolism, and the SAM is the methylation pool. So depending on this methylation pool, this reaction driven whether it drives till MMA or it goes to DMA, that results in different toxicity. If it goes till MMA and cannot reach up to DMA, then toxicity is more, and those individuals having more MMA, that is incomplete metabolism, due to maybe the inefficiency of S3MT or maybe the parent compound, that is the pool of MMA, all these things actually decide that what would be the fate. And here we have analyzed, we took the individual's urine sample, urinary concentration, and did ELISA on the sample, and we found that those, we are showing that the individuals who are having this arsenic-induced skin lesion, basically their MMA pool is reducing. So MMA pool is one of the very important driver and the S3MT enzyme in this entire story. So here we have shown that S3MT that converts to SAM to SAH, MMA through urinary transcription, and how this promoter methylation difference occurs. We studied this promoter methylation is a S3MT gene, and we found that due to this promoter hypermethylation, gene expression of S3MT is reduced and ultimate fate is different. We are working in this entire pathway in a lot of the metabolites. We have worked on the sample and also the connection with total MMA DMA in the urine. So now coming to the arsenic-induced DNA damage, here we found that apart from sample, arsenic that causes another important thing, that it does not only causes the DNA damage, at the same time it causes inhibition in the DNA repair enzyme. So we made two interesting experiments. In one individual, we studied that we took the blood samples from the arsenic exposed skin lesion, no skin lesion, and entirely unexposed population. We had their blood samples, took in the laboratory, and we made an arsenic exposure in the cells and found that what are the DNA damages are occurring. So, and thereafter, we also studied that what are the particular DNA repair enzymes and how are their expressions. In all these different kind of studies, our main objective was to identify whether during this skin lesion individual and no skin lesion individual having same arsenic exposure, whether their damage is same or their repair inefficiency or repair efficiency is different. So here we found that arsenic exposure causes significant amount of DNA damage, and here again that the individuals having different repair capacity makes the outcome difference. So always we can see that there is a balance between DNA damage, DNA repair. Those individuals, in a very simple way, that those individuals who are capable of maintaining this DNA repair system up compared to the DNA damage, they are getting in a better scenario compared to those who are not efficient in repairing. So we did it earlier, and nowadays we categorically chosen all of these DNA, all of these enzymes and this targeted gene MNH, MSH separately studied their promoter methylation just to understand that whether only the genetic consequences, genetic changes, only we are talking about any SNP or any mutation, whether that is only working, or the promoter methylation. That is, what is the contribution of the epigenetic perspective. And each of this, we find that yes, whether their promoter hypermethylation or promoter hypomethylation is working on. So we understand that the scenario is complex and complex, and only very little thing we understand. So each of these genes, we have studied this promoter methylation and their expression. We studied another thing. We were trying to understand, that is, what are these histone modification. Apart from this promoter methylation, what are these histone modifications are going on? To understand this, we had a panel. We took a panel of H3 where there were 24 specific modifications. And since we don't want to be biased, so we rely on this histone panel, and we gave the blood samples. We take the samples of arsenic exposed population and extracted the histone and studied that which are the specific modifications we are looking into. So here we found two important things. One is H3K79ME1. This is significantly upregulated, and H3K36ME3 is significantly downregulated. Since we don't have that enough fund to study each and every changes to cross-check or validate in our lab, so we took a bigger threshold. That is something beyond 1.5 or less than 0.5. So which are two drastic changes. We take it. First, we choose them and found that how they are going to change. So H3K79ME1, I'm not getting that slide here. That slide is missing. We find that H3K79ME1 that stays in the upstream of and it causes the DNA damage more. This is a DNA damage marker, and we found in those individuals, H3K79ME1 is more. In the arsenic skin lesion individual, we found their DNA damage is significantly more. Similarly, H3K36ME3 downregulation, and it was an indicator that DNA repair is also inhibited. We have studied a few mitochondrial modifications, changes which are going on. We have studied the D-loop methylation, ND4, ND6, MD mitochondrial DNA copy number, and found there are difference between skin lesion and no skin lesion. Ankita studied on the telomere integrity. We found that the telomere is one of the important part for the DNA damage maintenance, the chromosomal maintenance, and here we found that telomere, whenever we started our work, so far we have seen that telomere degradation or shortening of telomere is associated with cancer, but somehow we are finding in our experiment that telomere lengthening, we are observing telomere lengthening in our population. We took the blood sample as also the tissue sample from the arsenic exposed BCC patient. We are constantly looking for the telomere lengthening. However, it is not due to telomerase overexpression. So you are looking for that what is happening, and thereafter we found that this is the alternative telomere lengthening. This is the main driving force, and it's causing the lengthening of telomere. So ALT is happening in case of arsenic exposed population. We find that those individuals having urinary arsenic concentration is high, their telomere lengthening is also high. So telomere lengthening is another factor that we have identified, which is causing the difference between skin lesion and no skin lesion individual having similar arsenic exposure in the population. So these are the subtelomeric methylation patterns that we have studied. And this we have summarized that what are the things which are changes. And here we made another thing. We were working in arsenic exposed children, and our main objective so far we are working on the adult population more than 15 years of exposure, because arsenic says that up to 10 years it has a latency period. But our objective was to identify that whenever we are developing the skin lesions, palmar-plantar keratosis, and which are the pre-malignant lesions, then it's very difficult to make the population in a safer. So what are these early biomarker or epigenetic biomarker, if we can identify in the children population, or at the what age that we can identify that they are going to have this kind of problem. So for this, we took the population or the child from 5 to 15 years along with the adult population and compared the same parameter for the telomere length, the DNA damage, all these parameters we have checked between these small children and the adult population with the notion that which are the changes, the trend line it's showing at the early age, so that we can pick them at earlier age and can go for any protection. Any protection means that even if we can aware them or at least having safe alternative of water, because we have seen that if you have a long-term exposure, then it's not going to have any help. So better to identify them early, take care, or having some alternative. There are a lot of bioactives which are having beneficial role on arsenic, which are causing actually the antagonistic of arsenic. So that kind of food or antioxidant rich food, if we can provide them or identify them. So a lot to be done. Just only we have identified, yes, here it is that data that the children 5 to 16 years and adult 25 to 50 years from same village, and these parameters, these are the genes you can show here by the color code. With this color code, we have identified that the risk, this purple to light gray. So those individuals having more purple colors, that is this MLH, is very significantly it's showing hypermethylated. So we are trying to identify this pattern between the children and the adult population so that we can take some point of care. With this, I thank you, my team, because this I'm representing the entire work, mainly from the IICB and this my students. Thank you so much. Just one thing, that in our next session we are that's why we want to make a panel where these affected people, they can be on this stage. They can hear from the expert. In the next session, there will be a lot of speakers who are working in this arsenic in groundwater. Rashun Bhattacharya is there, Abhijit Mukherjee is there. Both of them are geologists. So in which panel, from in which layer arsenic is coming, they can tell on that. In the resource management, water management, Dr. Aruna Bhumajumdar is there. From environmentalist, Professor Aniruddha Mukherjee is there. And as participants, the contributory from Murshidabad, Habibul, who actually, with him, we made this villagers, we reached to the village people. So that person is coming, and his son, now he became doctor, he is also coming in the next session. The Nodia with whom we made this Munish Bathan and Nodia camp, where we have seen these people, they are still not having water, that I am emphasizing again and again, because this is a pain still in 2024 with whatever the research we are going, if it does not have any meaning or it does not have any social translation, we feel that we are failed. So that people, they are also coming. They will be sitting here along with all the scientists, all the experts here and on the dais, so that they can take some take-home message from the experts, so that they can give some suggestions that where we can move ahead. Only then we will have any solution for the environmental that will be sustainable and that will make our society really survivable. Thank you.
Yeah, likes. Thank you, Preeta, for this very important work and impressive presentation. So I think there's time for a couple of questions.
I have a very silly question. As we age, our telomere gets short. Data shows that it's also a telomere gets lengthened. So arsenic should prolong one's life.
That's a thing, sir. Yes, in arsenic we find, I don't know what it is, we find a lot of opposite things. Initially, when we working, we find that apoptosis is going significantly higher in arsenic exposed population. Nilanjana is here, and Nilanjana did a lot of work in immunology, and we find that arsenic exposed population, apoptosis is going in a very good number. So we thought that yes, then it's very good because it's identifying this defective cell and going to the program there. But we found that it's accelerating apoptosis is going on. So that's not right thing. So for in telomere also, we find that telomere lengthening, we find that a lot of telomere lengthening is also leading to cancer. So it's absolutely depending on the balance. Not too much of telomere lengthening and not too much shortening. Both are leading to cancer by different pathways. That's why the pathways are not very simplistic, as you all are experts over here, and all these are connecting with a number of other hubs, and we find that these balance, the DNA damage repair pathway and DNA damage response, or this telomere, everything, that critical balance is the important thing, which will make our lives safe.
Research for a very, very long time.
Any questions from the audience?
Yeah, there's another question.
Yeah, I stop. So I have a question. So do you have any plan for doing transcriptome studies with these blood RNA with the non-exposure people, with the exposure people, as well as the methylation study, whole genome bisulfite sequencing? Do you have a plan to do that? Then you can connect with your objective with this very clearly, right? Okay, thank you.
We have done both the microarray study as also and recently we have sent for an NGS. I was not prepared today to show all those data. The microarray data is already done, and from that queue we are taking all these genes. So it's not any of the genes were selected by an individual. So we had those entire data, transcriptome data, and NGS data also, from which we picked up the genes.
Thank you.
So if there are no more questions, I thank Rita for a wonderful talk and helping us to continue the session in absence of the other speakers. Thank you so much.
The entire team, because I am presenting only on behalf of my lab and the previous lab altogether. So this
Now we'll be proceeding for a lunch, and please do come back by 2 p.m. for the next technical session. Thank you, chairs, for doing the honors. Thank you guys so much. The posters for the poster presenters, there's an announcement. The room, a room on the first floor will be opened at around 2 p.m. So you can go there, put your poster up according to your poster numbers, and come back to join the session. Thank you. আপনারা আবার প্লেলিস্টে একটা ছবি